Good morning, everyone. Welcome to the first session. Uh, Projects funded by the NRM NIH Extramural Program. And let's kick off this wonderful workshop to hear some research focusing on computational digital curation, ranging from metadata curation, clinical imaging labeling and analysis, phenotyping, cardiovascular and COVID research. So these works are funded by the Extramural Program at the National Library of Medicine, NLM, and other NIH institutes under multiple programs. And among these work, a couple of them are supported by the lately developed program at NLM through grants under the computational approaches to curation at scale for biomedical research assets. I will post this NIRM funding opportunity, which is active announcement in the chat, so you can take a look, and you are very welcome to look into this program and send your application if you are interested in the research goal supported by this program. And so this NIRM program supports novel informatics and data science approaches to accelerate the availability of and access to secure, complete data sets and computational models and tools that can serve as the basis of transformative discoveries by improving the speed and scope of the curation process with innovative computational approaches. I'm very excited that we have invited 5 wonderful speakers with us today. So with a pleasure, I'm going to introduce our first speaker, Dr. Mark Musin. Dr. Musin's presentation title is Technology is Not Enough. Curation at Scale Will Require Mobilizing Scientific Communities at Scale. So just to give a very brief introduction for Dr. Musin. Dr. Musin is a professor of medicine and biomedical data science at Stanford University, where he is the director of the Stanford Center for Biomedical Informatics Research. He's known for his studies on the use of semantic technology in biomedicine and has been the principal investigator of several projects that have led to widely used systems such as the BioPortal. And he's currently supported by the NLM on a project known as the Metadata Power Wash, which is exploring how to analyze metadata from datasets in existing repositories and to cover this meta to more standards adherent forms. This is what Dr. Musin is going to talk at today. So with that, I would like to turn the floor to Dr. Musin. So Dr. Mark Musin, so you can set up your slide.
Sure, I think I'm gonna need to have the power to share my screen.
Right, so.
There we go.
Great, thank you. And so for audience, so please, so you are very welcome to post your question. Please use the QA feature and to post your question. So we will try to get your question as many as possible. However, given the time limitation, we may ask the speaker to respond through that feature at a later time. Dr. Mark, so please start, thank you.
Thank you, Yanli, and thank you for asking me to join you this morning, and thanks to all my friends and colleagues whom I see in the audience. What I'm going to do in the next very short period of time is to give you an overview of some of the technology that we're developing at Stanford to help us with the curation problem, but also to leave you with the idea, basically reflecting on comments Dr. Brennan made, that technology is certainly not enough, and that for us to be successful and be able to curate at least data at scale, we're going to need to mobilize scientific communities, funders, publishers, and everyone involved in the scientific enterprise. And I'll talk more about that in a moment. Let me start by at least getting attention to the elephant in the room, which are the FAIR principles, which have already been mentioned a couple of times, and we haven't even begun the sessions yet. The past 5 years certainly have been a period of time when people have really been focused on the importance of making data findable, accessible, interoperable, and reusable. And although I'm not going to lead you through the FAIR principles, I want to make one point. When you look at the list of the FAIR principles, which I'm sure all of you have memorized carefully, I'm kidding, of course, what comes to mind is that almost all the principles deal with metadata. And if you take away the FAIR principles that actually deal with characteristics of data repositories, which investigators really have no control over at all, what you realize is that what's left are principles that deal with the metadata that investigators author, and indeed requirements that those metadata be rich, that they adhere to community standards, that they be domain relevant, all kinds of things which obviously are not really computable, but absolutely essential for making metadata available so that data sets can be fair. And the problem, of course, as we know, I'm not going to go into detail here, is that when you look at existing data sets online, most of the metadata are frankly terrible. And so our goal in the work that we're doing at Stanford is to make metadata richer and more standardized so our data sets can be more fair. And that means if we want to have fair data, what we ultimately need are the following things. We need ontologies that will provide controlled terms for the data that rather the metadata that are going to describe our data sets. We need reporting guidelines that give us a standardized structure for the metadata components, reporting guidelines like Miami and all the others that define attribute value pairs that provide a mechanism for making sure our metadata are always going to be searchable. Technology, which I'll talk a lot about, that make it easy to author good metadata in the first place. And I think most important and usually overlooked are the procedures that we need to create community-based standards in the first place, because this is really what is most essential for our technology to have any durable effect on improving the fairness and usability of the data that come out of scientific research. Let me start with the idea of ontologies. We know that's kind of intuitive. But when you look at what at metadata online, you see, metadata are not good. These are taken from Geo. This is all the different ways in which you can express the age of the subject in the Gene Expression Omnibus, the NCBI repository for data that are related to gene microarrays. And what you see is if you want to search for age in Geo, you don't search for just AGE. You have to figure out all the possible ways in which investigators may have described age. You have to know all the possible typos that they may have entered into their metadata. And frankly, it becomes a real difficult process in order to find things that are necessary, because unless investigators incorporate ontologies and standard value sets into their metadata, our metadata are never going to be fair. We at Stanford have created a resource called BioPortal, which contains all of the biomedical ontologies in the world, basically, at least those that are available openly. And we use BioPortal in a number of our projects, and I'll come back here in a moment. I said we need reporting guidelines. Reporting guidelines basically give us standard attribute value pairs for talking about the metadata that will ultimately give us fair data. And when we think about reporting guidelines, that sounds like an obscure notion, but it's not, because we're all pretty much familiar with these ideas. They started more than 20 years ago when publishers said, we can't publish 25,000 genes in a PDF publication. We need to have gene microarray data online. And we have to have a way of describing it that will allow readers and scientists to be able to access those data in a reasonable way. So the microarray community really took up the mantle and said, look, we need to be able to have a standardized mechanism for describing what was the experiment, what was its substrate, what array platform was used, what were the experimental conditions. And the result was Miami, which has become really one of the poster children, if you will, for how scientists can standardize the way in which they talk about their work. And certainly in biology, it didn't stop with Miami. We have MIATA, we have Miriam, we have Minimesh, we have MISFISHI, and over 100 reporting guidelines that the Fair Sharing resource at the University of Oxford manages and allows us to review and to incorporate into our work. These are exactly the kinds of community standards that we need to use to structure metadata. And again, to emphasize another point that Dr. Brennan made in her opening remarks, standards are not the domain of arcane scientists who sit in small rooms. This is the business of everybody in science, because all of us need these kinds of science, all these kinds of standards for our work to be successful and for our data to be shared. Now, I mentioned ontologies and reporting guidelines, but obviously I would not be a technologist without having a chance to tell you about some of the technology that my large team at Stanford has been working on. I don't have time to talk about these projects in detail, but I'm going to mention the CEDAR Workbench, which was technology we developed under the NIH BD2K program, which is an editor that allows researchers to create standards compliant metadata from ontologies, CDEs, clinical common data elements, and reporting guidelines. New work that we're doing called the Metadata Power Wash, which is an R01 funded by the National Library of Medicine, which fundamentally gives us a spell checker for metadata that allows us to take legacy metadata, like those ugly metadata you saw from Geo, and attempt to convert them to a standards adherent form to make the underlying data sets more fair. We'll talk about the Metadata Power Wash in a moment. And finally, the Fairware Workbench, which is technology being funded by the Research on Research Institute, largely through the Wellcome Trust in the UK, that basically builds on the technology that's being created through the Metadata Power Wash and provides a mechanism whereby funders can look at the metadata being created by their grantees, make assessments about how good those metadata are, how fair the underlying data might be, and basically determine what are the community standards that are most successful in allowing scientists to create fair data sets. Let's start with CEDAR. CEDAR, as I said, was supported by the Big Data to Knowledge Initiative at NIH, and it creates a library of reporting guidelines. And people who use CEDAR are able to create new reporting guideline descriptions, are able to use reporting guideline descriptions encoded as templates that have been produced by other investigators and share pieces and parts of these descriptions among themselves. We want to look at, for example, what do we have in this library as a way of describing the metadata for a biosample, one of the NCBI repositories dealing with human data. We click on populate, we can see what the template looks like. So here is a template in CEDAR describing the reporting guideline that captures what needs to say about a sample in order for those metadata to be stored in the biosample repository. Things like the sample name and the organism and the tissue and the sex of the subject and so on. What CEDAR offers is a way of filling in this template, making sure that all the data types are correct. And most important, when something like tissue is to be filled in with a value that comes from an ontology, CEDAR is able to go out to that bioportal resource that I mentioned earlier and give you a drop-down list of all the possible values that might be selected from that ontology that would be appropriate for this form. And more important, because we maintain a cache of all the previously entered metadata, we can identify what are the more likely entries that would be going into this particular slot based on what previous entries have been already entered into the template. And we know if the sample name is irrelevant, but if the organism is Homo sapiens, then by far and away, most of the biosample metadata describe the tissue being from blood, and that's at the top of the list. Liver is next, and so on. And so basically, we can sort the entries in all of our drop-down lists to make it really easy to navigate large ontologies and instead see right at the top of the list what's going to be most relevant and fill in these forms really quickly. So CEDAR gives you the ability to use standards like ontologies and CDEs in order to create metadata that are compliant with community standards. The other technology I want to tell you about is the Metadata Power Wash, which is technology that's being created under a new R01 from NLM, which has the ability to convert metadata, legacy metadata, junky metadata in bulk into a form which we think is most likely to be standards adherent. We can't guarantee the results are going to be perfect, but we think of the Metadata Power Wash as something like a spell checker for metadata. And so the input is going to be a set of metadata records. There may optionally be a CEDAR template, which gives you a reporting guideline that might have been considered the basic structure for these records, but if not, the Power Wash will figure out what reporting guideline might be most appropriate. And the output is going to be a set of metadata records which represent basically the spell-checked metadata. If we were to take the existing metadata and try to find the best way of representing them using existing standards, these would be the corresponding metadata values. And we would have a report describing exactly what we did. And although there's not time this morning to go through the details of how this all is working, fundamentally, you can see that we take the existing metadata records one by one. We see how their fields align with existing templates that are in CEDAR to align them with reporting guidelines that might be used or might conceivably be used. We see if we find a reporting guideline that's appropriate, whether there are any fields that are missing. We see if there are missing values for those fields. We try to see if the field names need to be standardized, and we see if we need to standardize those field values, usually using ontology terms from BioPortal to ensure that we can make good guesses at what those values should be. And ultimately, we generate what we consider to be repaired metadata and store those repaired metadata in CEDAR, not to replace the metadata from the original repository, but to provide an alternative repository of metadata that users can search, hoping to find mechanisms for locating metadata and corresponding data sets in the original repositories, knowing that the refined metadata will be more searchable than the legacy metadata, which may not adhere to standards. So our goal here is simply to be able to increase the fairness of online data by providing an alternative set of enhanced metadata that users can search. This underlying technology actually is being adapted for a technology which we call the Fairware Workbench. This is work that is being supported by the Research on Research Institute and basically is viewed as technology that's going to help funders to use the metadata power wash to assess how well their grantees are using existing standards, and fundamentally where those standards might need to be improved because they might show difficulty in how investigators use them. What I'm going to show you now is not a real system. These are wireframes that we're using right now as we mock up and ultimately build the interface that will allow the underlying metadata power wash to help funders do this kind of work. But fundamentally, in this technology, users will be able to enter the DOIs for the data sets that they care about. They'll say, let's search those DOIs, and they'll get a list of DOIs or rather metadata that relate to those DOI pointers. And we can, if we want to, then evaluate whether those particular metadata records adhere to standards. And what we get back is a report saying, for example, that this particular metadata descriptor has 26 issues. And we can actually look at those if we like. We can go to the report and see that for this particular metadata record, there are 9 errors and 17 warnings. This is the list view, which is actually kind of hard to read. If you go to the metadata view, which we think is going to work out better, you can see that for this metadata entry, there's a title. By the way, this is the template for the DataCite metadata schema. There's a title, there's a language, there's a publication year, and so on. And Fairware is saying through the power wash, look, the language is ENG. That's not a standard value. You want to replace ENG by, for example, US English. January 2014 is not a valid publication year. You probably want to replace that by 2014. Documentation is not a topic that's in any kind of standard terminology. Maybe you want to replace it by the corresponding ontology entry in BibFrame, and the user has the option of accepting or declining those recommendations. Ultimately, what the power wash gives us is fundamentally what the funders want to see, which is a report that will say, for the records that were entered, how many were valid, how many were invalid, what were the kinds of problems that people found in using these standards. And the goal here is to be able to use this Fairware tool as the basis for making recommendations to standards bodies as well as to grantees about how best to make their metadata fair to improve standards and standards that might be easier to use in future versions. So the CEDAR Workbench basically gives you the editor for making sure that entries are standards compliant at the time that an author creates new metadata. The metadata power wash in bulk is our spell checker for legacy metadata. And the Fairware Workbench is future technology that's going to allow funders to be able to evaluate legacy metadata and identify where there may be issues that could hopefully in the future lead to better and more fair data online. But technology is not enough. We need the standards in the first place. And again, as Dr. Brennan said, standards are not the domain of arcane specialists. Standards are everybody's business. And over the past few years, we've been working with the GoFair Foundation to develop what we call metadata for machine workshops, which allow us to work together with scientific communities in order to create standards that ultimately can make data sets more fair. It was very exciting to us that these workshops are very easy to put on, relatively efficient, and have led to a host of new standards, probably the most important of which are standards that have been developed by the Netherlands Organization for Health Research and Development, which basically has given us the ability to use standards created from these M4M workshops to ensure that all of the data sets created through the RFAs that are issued by this funding organization in Holland comply with community standards. And it's basically a funder putting on workshops, getting communities mobilized to create the standards that will ensure that their data sets are fair, and ultimately using the technology that I've just described to ensure that those standards are adhered to. So that means technology like CEDAR and the metadata power wash to clean up metadata and ensure that metadata are fair in the first place. And ultimately, what we see is an issue where this kind of technology will be very valuable in ensuring the fairness of online data and making data more useful. But probably the most important part of what I've said today is that this notion on the part of funders, publishers, and professional societies to stimulate the creation of the standards in the first place is really where the world needs to be heading. And where I'm really most excited about where we can get together both as grantees and as funders, as technology developers, and as members of professional societies to create the community standards that will allow the kinds of technology that are being funded by the NLM to make our data sets more fair and more usable. And let me stop there and thank you.
Thank you so much, Dr. Magnuson. So we have time only for one question. So let me read a question to you, Dr. Magnuson. So can CEDAR help alert the ontology developers when no suitable term exists in bioportal? A short answer would be appreciated.
Sure, thank you for that question. And the answer is we are working on exactly that kind of technology. For the past few years, again, with support from the NLM, Stefan Schurer, who's on the call, has worked with our group to create a system called OntoloBridge, which allows the bioportal system to easily communicate with the developers of certain ontologies to let them know when there may be errors or maybe enhancements that should be made. And right now, we've actually taken that kind of capability, moved it into CEDAR, so that as investigators use ontologies in CEDAR to author new templates, or when they fill out templates and see terms that might be missing, they can actually make a notation which basically gets communicated back to bioportal, which can use OntoloBridge to make those kinds of notifications. I think the bigger issue is what we really want are ecosystems that bring together technologies like CEDAR, like bioportal, like OntoloBridge, like the metadata power wash, that provide a uniform foundation for allowing users to create healthy and standards adherent metadata and to use those metadata in their work for the purpose of re-exploring existing data sets.
Yeah.
We're moving in that direction.
Thank you so much. So we have to move on. Thank you. So Dr. Musa, there are other questions. You can continue to respond through the Q&A. So Dr. Stefan Schurer, so please set up your slide and I will introduce you. So our second speaker will be Dr. Stefan Schurer. His presentation title is Novel Tools to Enable Fair and Open Data. So Dr. Stefan Schurer is a professor of pharmacology and associate director of data science at the Sylvester Comprehensive Cancer Center and director of digital drug discovery at the Institute for Data Science and Computing at the University of Miami Medical School. Dr. Schurer serves as a principal investigator in the National Research Consortium Library of Integrated Network-Based Cellular Signatures, the LINCS, and illuminating the druggable genome, rapid acceleration of diagnostic, the RADx, NIH, and previously Big Data to Knowledge, BD2K, and the Molecular Library Program. So in his in this workshop, he will illustrate some of the tools and infrastructure they have built in this project to enable fair and open data. So Dr. Stefan Schur, please take it away. So please, so we have 15 minutes allocated to your presentation. I will remind you when time comes up. Thank you.
Yeah, thank you very much, Andy, for the introduction, and thank you for having this amazing workshop and for inviting me. So I will walk you through a couple of projects we've been involved in and how we address and how we tackle the metadata challenge that Mark Moosen so greatly introduced. So here I want to acknowledge first my amazing team and highlight some of the collaborators that includes Barry from CDD and his team, Mark Moosen and his team, to do our prayer, and if you have time, Helena from university here. And touching on some projects, the LINCS project, IDG project, and like Mark mentioned, the NLM OntoloBridge project. If you have time, a little bit about RADx COVID SARS-CoV-2 surveillance. So starting with one project, the Library of Integrated Network by Cellular Signatures is a large-scale profiling, cellular profiling of perturbations with various different readouts, transcriptomics, proteomics, and so on. And we were the data coordination center for this project, so challenged with putting all of this information together. So what we did is, in a typical way, develop metadata standards for reagents, various different reagents, cell lines, iPSCs, antibodies, small molecules, and so on. What you can see here is the number of fields without much detail. This work is published that we formally described and then captured for those. The same thing, like Mark also mentioned, for experimental metadata. Those includes, again, cells, nucleic acid reagents, proteins, small molecules, and so on. And then also the assay metadata. So what is important about the various different assays, ATAC-seq, RNA-seq imaging, proteomics, and so on. Capturing those. And then in addition, the data set packages, these include the actual data sets that are being generated and the metadata of all the reagents and describing those as well. And then what we built is the processing pipelines to allow data submission via submission tools, via APIs and or via FTP servers. This was at this point a semi-automated process to capture all the reagent metadata, assay metadata, like I mentioned, data set metadata, experimental, and also the processing pipeline and the data files, followed by validation that includes terminologies, completeness of the content, mapping the fields, and then the pipeline validation. This was computationally done, of course, in a manual way by reproducing the pipelines, and followed by registration, building a registry for all the reagents. And then being able to link this via identifiers to ontologies and other external resources, package the data together, QC it, and then provide access and analysis capabilities to all of this data, metadata via the LINCS data portal that includes the data sets. And then also in the second iteration, the signatures that then become computable. So showing you this LINCS data portal here, you see the various different data sets that are organized by different categories, methods, by the centers, projects, biological processes. For example, here, what are the general areas of assays, binding assays, kinome scan, and so on. And then you see here small molecules, cell lines, and so on that are also captured. Now it allows us to look at all the data sets and all the associated metadata that are linked here in the various tabs, metadata, download the data, and so on. And this data then becomes citable via unique identifier. And the same thing for all the metadata that are associated with any data set. Also, all these have unique identifiers. And it also keeps all the data together, the metadata and the data sets and the different data levels from raw data, processed data set, all the way to, I go back here, all the way to the signature level. And these can also be individually cited. So at this point, lots of these tools are very specialized, and some of the processes were manual. And then in another project, Illuminating the Druggable Genome, we were able to improve this and formalize all of this a little bit more. The IDG project is really about prioritizing understudy targets of likely druggable protein families for ultimately drug development. It's a large-scale consortium similar to LINCS. And how it's organized is we have the data generation, data and resource generating centers, a knowledge management center, and a resource dissemination outreach center to build the technologies to capture all the information, and various collaborations, and there are various other peripheral centers and projects involved with this. But quickly going to the topic here of describing resources as metadata. Examples here for this project with antibodies, cells, genetic construct, mouse, peptides, small molecules. So we developed the metadata standards in a much more efficient way than we did it at Links, make those available via the website. They're also, of course, available via fair sharing. And now we build a process to describe all the resources in a descriptive way, to essentially describe what are the types of standards, the types of information we want to capture, so that Mark mentioned the reporting guidelines, what type of ontologies we use to describe each of those. And then we build tools to now make those descriptions that typically researchers will generate in some sort of tabular format into a JSON schema. And then when this data is submitted, to make those all these resource description available as JSON LD in a Mongo database that then can be accessed, for example, via a smart API. So those tools are available, and we built this resource submission system. This system cross-authenticates with the CEDAR technologies that Mark already mentioned, because it integrates with CEDAR. And if you load onto this page, you see a landing page, and you see the various different material resources called battery agents, or various different dataset resources or dry resources that we can describe. And then also it's possible to, of course, review all the information and look at this. And if we, for example, look at the material resources, we can see a genetic construct, peptide, and so on, all of those, and then the various different data sets that can be described. And any of those can now be submitted, and it can be done via either a data file that then can be mapped to the metadata standards that have been previously formalized, like I mentioned, or it can also be done via a form, and those are the CEDAR forms that Mark mentioned already. And there the information can be captured in a similar way, like importantly allowing controlled vocabularies via ontologies and so on. So what we found is people like to batch submit information, but then they like to use the CEDAR templates really to review or to make corrections, or to for example correct something. Now all of this information then is made accessible to the Pharos IDG knowledge portal, which is available here, and also via the IDG website druggablegenome.net. So briefly, what I want to change subject here and talk about development of resources like ontologies. So if you think about like drug discovery, the development of compounds and identifying those compounds in the first place, often compounds are identified that are artifactual, and there are various different mechanisms like redox cyclers, covalent modifiers, metal complexes, reactive compounds, unstable compounds, and so on, that are shown here in an older paper. And now if you look at the MLP program, the library was managed at the end at Evotec. Here you see what kind of compounds were ordered very frequently as follow-up from primary screens. And you see many of those reactive compounds were ordered over and over again, and it's likely an artifact. So the lack of organization of and annotations of assays at the time in the MLP led us to develop bioassay ontology. So bioassay ontology is now a description of those drug screening-based assays. And the important feature of it that it orthogonally describes various different categories that are important for those various chemical biology assays. For example, what is the assay format, like is it biochemical, cell-based, and so on, using terminology that people use in this domain. What is the assay method? So how is a perturbation translated into detectable signal? What is the actual endpoint that's generated? What is the physical method and with which a signal is detected? Now, bioassay ontology is now, after has started development for many years, widely used in many pharmaceutical companies, many different projects. More recently, we work with all the large pharmaceutical companies. This is very driven by the Pistoia Alliance. And they have decided this is going to be the standard they're using for assays. And we work with them to expand it and develop it further, and also contract research organizations like Eurofin are now working with us to expand this. So it's great to have to see this project evolved. to this level. But what I want to briefly explain is what the point of it is, just illustrate how this can be useful. So for example, if we look at various different assays like enzyme, kinase activity assay, or reporter gene assays, or cell viability assays, they all have different names, but they all work in the same way. They all use the luciferase reaction. Luciferin and ATP goes to oxyluciferin, and then emitting by chemiluminescence. And now bioassay ontology allows us to describe all of these assays that have different names, and they have different methods, different called methods that you can see here, and describe them now in a formal way so that we know they all kind of work in the same way. Now, if we know they work all in the same way, what we can do is now identify what are the assays that work in a similar way, and we can now look for the individual compounds and how many assays each compound is active versus inactive. We can see here there's different assays on the bottom, and then if we look for all of these compounds, we can see 2 of those clusters and all of those that have very many actives in the luciferase reporter gene assays, kinase activity assays, or luciferin coupled tox assays. And you see the compounds that come up here are luciferase stabilizers, or we can see those ones that are very frequently showing up in reporter gene assays or cell viability assays. And you see these are cytotoxic compounds, right, that are intercalators or reactive compounds like shown here. So it's illustrating the utility. If we have this assays annotated, we can do better analysis, like Patty mentioned in the beginning. So now we are expanding the assays with those collaborators, like I mentioned, into pharmacodynamics and pharmacokinetics assays. This will be released very soon. You see a Sentry view of those different types of assays. Now, like Mark mentioned, what we have done with support from the NLM, developed a system, OntoloBridge, which is available on ontolobridge.ccs.miami.edu. And it addresses the challenge that if an ontology is used and the term is not available, to quickly be able to request the term. So we built the system, we implemented it here as a standalone website, but it's also, like Mark mentioned, implemented in the BioPortal. So if you look at an ontology, now you can go here, and if you're logged in, to see for several ontologies the new term request. So you can add the new term, a description, and then this gets submitted to the system and is available. Now, it was also implemented in the CDD Collaborative Drug Discovery Bioassay Express tool, which is now BioHarmony. So this is a semi-automated curation tool that takes text and then identifies certain terms that with which this text should be annotated. And then it does it in it prioritizes terms using machine learning, and then lets the expert choose among the top terms what is the correct one. So this is an annotation interface. This is available at bioassayexpress.com. I will show it in a second. So you can do those annotations in the system, and then if it's needed, request a new term, add a label for this new term, add a description, and then also has an administrative component for an administrator to review all the requests and then approve, and then they get submitted to the system. So now that tool has been used to annotate close to 4,000 assays in PubChem, and you can find them here and in the bioassayexpress.com tool. You see just a few examples here. So I still have like 1 more minute. So I just want to briefly mention that I'm now very happy that we were able to use the tools that we developed and the experience we have gained in a SARS-CoV-2 wastewater surveillance project that is part of the RADx-RAD program of NIH. And basically what we do here is take wastewater samples and also do human surveillance and capture all this information, and then enable the analysis of wastewater and then integrate all of this data, and then be able to characterize outbreaks and make predictions that can then do policy decisions. So I don't have time to go into all the details. This is another talk, but I wanted to mention it because Paddy mentioned it in the very beginning. So how important it is to leverage data management practices, metadata management in those areas, and we were able to do it here. So with this, I will stop to stay on time and want to thank you all again for the for the workshop and for inviting me to speak. Thank you.
Thank you so much, Dr. Stefan Schroer. So there are no questions posted, but let me ask one, and then and I just please give you can comment quickly. So if you are going to give us sharing your experience using CIDR, so what will be that?
Yeah, so we worked with Mark already in the links project using CIDR, and at that time. We actually developed together the platform to allow us to capture information in links. What was challenging is that in links we wanted to really put together different types of metadata together with all the different data sets and to group them all together in one set. There was a limitation in CIDR at the time, but we still were able to use it. But ultimately we had some set of tools that was very specialized. We didn't have like a very generic type of tool, and then later on in the IDG, in this RSS system, we really integrated CIDR completely, right? And the schemas are compatible, and people can go to CIDR, or they can go to RSS, or they can map the files, and it's all going to end up being exactly the same thing, right? So you can submit it in one way, and then you can look at CIDR and make corrections, and it works well together.
Thank you. Yeah, that's wonderful. Thank you so much. And there are questions posted, so in the QA, so you can just respond. So Dr. Dexter and Hadley, please set up your presentation. Let me introduce our third speaker, Dr. Dexter Hadley. So his presentation is From Curation to Community-Driven Artificial Intelligence. So Dr. Hadley expertise is in translating big data into precision medicine and digital health. He earned his PhD in genomics and computational biology while at medical school at Penn, and he trained in clinical pathology while at residence at Stanford. So he spent 4 years at UCSF as a faculty member, where he won several NIH grants and developed his research program. As a new faculty member of the University of Central Florida College of Medicine, he is pioneering a new division of artificial intelligence to leverage the transformative power of AI from medical school through clinical practice. So in general, the endpoint of his work is rapid proofs of concept clinical trials in humans that translate into better patient outcomes and reduce morbidity and mortality across the spectrum of disease. So Dr. Hadley, please take it away. Thank you.
Awesome. Can everybody hear me? Thank you, Yan Li. Thank you for inviting me to this long overdue workshop, and it's nice to see all my friends. Disclosures. Yes, as you mentioned, I spent 4 years at UCSF where I won this award. Initially this was sort of, the idea was we're gonna curate mammographic imaging. Here's all the digital imaging in the Department of Radiology at UCSF. Until I was there, I left a few years after this. But there were about 5 million records in the EHR system there. Across, everybody in the ER gets a chest X-ray. There's a ton of obviously chest X-rays. I want you to focus on the yellow, which is mammography, because there are about 800,000 mammographic images at UCSF that were available for me to analyze at the time for breast cancer. Those who know about breast cancer know that mammography is not as effective as we thought it was. In the 80s, Congress mandated mammographic screenings, one of the only diseases that the FDA regulates. And what you can see from this graph, which is the size of tumor excised in surgery, if mammography did work, you'd expect large and metastatic tumors to decrease in size. It is not. But small and sort of incidental tumors have increased this in situ, ductal carcinoma in situ. So, we're not catching the disease as early in many of the population is what this shows. So mammography has some there is room for improvement. And the reason is, most of the people reading these scans are fresh out of residency, and this is a two-dimensional mammographic scan. There's two views of two breasts, and it's very difficult to catch pre-cancer on this. So we thought maybe we can train deep learning algorithms to, we had all this data, almost 1 million mammograms. We thought we could train deep learning algorithms to reduce this inefficiency sort of in the diagnosis. The sensitivity specificity is, 80–90% is pretty high, but so many women, the guidelines for screening, the FDA approved guidelines, so many women go through screening, the false positive rates are very high. So there's room for improvement. So here's all the data that's sat in UCSF's electronic health record system. We had about 1 million DICOM images. These are the exact files. We had about, this was indexed by 166,000 different reports in the radiology database. So this is in the PACS system, this is in the EHR system. And then there's another COPATH system with about 33,000 pathology reports. So we had to curate this entire stack of clinical data. And obviously we did it computationally. So here's what a pathology note, the nice thing about cancer is there's a gold standard. I trained at Stanford in pathology, and this is the gold standard for diagnosis. So we had to read about 33,000 of these pathology notes. You can read it here if you want, but here's, and the idea is we use natural language processing to read these. We trained about, I think we read about 2,000, about a tenth of them manually. And this note would be the left breast is positive because you see invasive lobular carcinoma here. There's no pathology for cancer in the right breast. So this is kind of what the image would be. Remember that the FDA regulates everything about breast cancer. So with very little sophistication, let's say, just the plain old logistic regression, we were able to predict almost complete accuracy in the left, the right, the positive, and the negative for breast cancer. We published that. Not to beleaguer the point, but if we're going to train a deep learning algorithm on routine clinical data, which is what we propose to do, you're going to find defects in the imaging. And the medical system is such that nobody tracks these defects, to be honest. There's fields within the DICOM file standard, but it hardly identifies images like this, which is to calibrate the machine, hardly useful for machine learning. So we trained image curation to curate out such special images that are just not useful for machine learning. We published that. So now we can read text at scale, real routine text, and we could curate images at scale. So, we can essentially walk patients through the standardized workflows and sort of, this is a typical workflow for a woman, so January 1st, you get a bilateral screener. Come back in February because the left is suspicious. You get a diagnostic. There's a fine needle. There's a lumpectomy. If we can read all these notes, which we can do, we can go back and label the images, which is what we did do. And I took all the images that I had and labeled them as such. Over here, you have the pathology. We have about almost 800,000 individual images. We labeled most of them negative and positive based on pathology. Over here, you have the BI-RADS score, which is another FDA-approved grading scheme of how much cancer the radiologist thinks they have. It's what probability of cancer. And we were able to very cleanly organize all of this data for machine learning. We're not claiming that the problem became we had too much data. We had 800,000 images and a lot of data to sort of train. So we ran out of computational power, but suffice it to say, we checked these curations with the Breast Cancer Surveillance Consortium data. We tested this. So here's 8,000 images that we labeled automatically. We tested in a machine learning algorithm for cancer, not cancer. Obviously it learned. And argument is we have quite a lot of data. Obviously, it's not just cancer and not cancer. There's a whole ontology, to Mark Muson's point, of pathology, and we labeled that and curated that too. So there's about 90,000 pathology, representative pathology slides in UCSF and distributed as such. So, the next level or phase of that work would have been to go scan those digital pathology slides and then make predictions, about drug response or et cetera. But then I moved. So about 3 months before COVID, I moved back to Florida. I'm from Florida. I took an appointment at the University of Central Florida. As opposed to UCSF, which has all of this data. UCF is about 10 years old. We have no data. We don't even have a hospital. The idea is, if we can read patients' imaging, we have no clinical data because we have no hospital. Can we just go ask patients? This is sort of to Patty Brennan's point in the community. Can we get their data? I'm here to tell you we probably can get their data because the standards exist. HL7, FHIR, DICOM, PACS. The companies exist. From my time in the Silicon Valley Health Gorilla, transmits EHR data between patients and doctors. Telluride transmits images. So we work with these companies to build up sort of the infrastructure to go into the community and try to get this data. And then COVID hit, literally 3 months before. Hiring freeze, couldn't hire any postdocs as a brand new faculty. And I was, involved in this research. And this research at the time was trying to show what everybody already knew. There was a huge disparity in outcomes of COVID. And if you remember, April 25th, 2020, Mike Pence had made an announcement about mandatory reporting. You know what Mike Pence didn't mandatorily report? Any ethnicity for COVID test results. Only 2 states, this is straight out of the CDC's website, April 25th, 2020, voluntarily reported ethnicity. So my colleague, Dr. Cyrus and I, were able to, show what everybody already knew without, obviously now CDC has data showing that New Orleans, Essex, New Jersey, Wayne, all of these were in the news. These are the counties on this axis. The blue is the African-Americanness or the blackness of the counties. And you have to sort of triangulate 2 different counties to show what is and has become quite clear. So we wondered, there was no way we were going to get any breast cancer traction in a pandemic. So we wondered if we could do something similar to what we had done at UCSF. I wondered if I could do something similar for COVID. I go to the public data for COVID, brand new pandemic in April 2020, or whenever this was. And the data just is absolutely useless, right? So, we want to predict who's going to be on oxygen, who wants to go to the ICU. The public data at the time just was, to Mark Meeson's point, the metadata was very useless. Nobody was really trying to predict in a pandemic what type of viral infection one had. But if you took this data, put it into any random machine learning, you get nice outcomes for useless metadata. UCF is, like I said, a brand new medical school. We have this focused inquiry and research experience. You're supposed to mentor med students into doing community research. We're in a pandemic now, and there is no community research, so we thought this would be a good opportunity to sort of take these ideas we had learned for breast cancer, see how many people would give us their images for COVID. So we launched this study, covidimaging.us, 2 very talented med students got the IRB approved. We built sort of this platform, and the idea is, we can go directly to the patient, ask questions about whatever at the dawn of this pandemic. In return, it's not breast cancer, it was a brand new disease. We didn't know anything about COVID. We didn't even know about long COVID at that time. But the idea was, can we return patients their image and ask them questions about their experience with COVID? So we launched and investigate the procedure of setting up such a digital community among patients. We launched Facebook ads to that effect to publicize our study. We had enrollment criteria. You had to be an adult and diagnosed with COVID and had an X-ray to work up that diagnosis. And obviously this is human subjects research now, a little bit different to the work I had done before and sort of throughout my career, but we learned a lot about how human subjects research on the internet works in a pandemic. So this was the first form you had to fill out. You just had to have these 3 criteria. 508 people over the 3 or 4 months or 5 or 6 months around the study replied. And as you can see, the informed consent, the HIPAA release, and finally where they had their sites, huge drop-offs in just the dynamics of recruiting patients online for these kind of studies. Needless to say, nonetheless, we got 48 imaging sites all over America. We were able to get informed consent and HIPAA release to go get the images. we're still in the process of getting those images. It cost $7,000 on Facebook ads to get those images during a pandemic when you really couldn't hire, when there really was no other way to get this data. $7,000, and this kind of shows just the dynamics. Everybody clicks on that first form. Few people actually go through the 3 or 4 emails required to sign up. It just kind of shows how we spent that money. Again, the NIH sort of approved all of this. This was during the pandemic. So this worked out to about $163 per recruitment. Right, we spent $7,000. There's 1, 2, 3, 4 different forms. At the end, it's about $163 per site, right? And then we asked them what were their ethnicity, and gender. 90% of responses were from women, and white women at that. It doesn't really help COVID, right? Where we know there's huge disparities in the outcomes of COVID and things, and it kind of just shows how difficult it is to recruit patients to correct those, to study the discrepancies. If it's $163 to recruit one average person, 90% of which are white women, it costs about $9,000 in Facebook ads to recruit one Black woman, in essence, which, became quite clear. This is not new. Half of these talks, I'm sure, are going to be about genomics. In 2011, Bustamante et al. Showed 96% of genomic sequence was European derived. And more recently, let's just talk about breast cancer. 40% of outcomes are worse in African American women. So this really is a very difficult divide to bridge with any research, I think. And what we're working on now, and obviously what the NIH is working on, many of us saw these sort of funding announcements. I think it was 150 million or whatever in these sort of post-COVID, just sort of attempts to decrease this massive disparity in data and obviously in outcomes. So going back to this FIRE program, what I realized from this grant writing experience is what we're going to do is sort of, all of these universities, all the Stanfords, all the UCSFs, all the Harvards are going to get together and put together all of their data. That much is clear. They're going to, harmonize it and normalize it and build awesome models, I'm sure, that work very well within their populations. What I realized from moving to UCF, there's only community medicine in UCF. Mind you, we have a med school. There is no concept of academic medicine here because our med school is run by a very large community hospital. And, I can argue maybe 80% of America is like Central Florida, where there is no big academic center. So what I find myself doing now is we're really figuring out how to reduce these very, very massive costs of recruiting, getting diverse data to even just measure the bias in the data and the potential bias that any AI or any kind of machine learning could scale. We just don't even have that data. So this idea of, and not to mention the social determinants of health, what I find quite, what COVID has sort of crystallized, especially in an environment like this, is that these large scale data studies, and genomics is a great example. We don't consider the social determinants of health at all because we can't even get back to the patient to ask them questions. It's just large scale sort of analysis. So I'll end with this. We really want, and we've partnered with a number of foundations where we want to use this large curation at scale to start reaching sort of these disparate communities. If there's a large number of false positive results in breast cancer screening, for instance. Not one person has their mammogram sort of on their phone like they should. There's no reason, there's nowhere for women to post sort of these, or any patients to post their data in a responsible fashion to get support from either their peers, other people going through this process, or clinicians who may be interested, right? There's no room on Facebook or Instagram to post mammograms. Obviously, that's a bad idea. So what we really want to do is build sort of digital communities of patients that have access to their data. DICOM, PACS, HL7, FIRE, and OMOP are all existing standards that make this quite doable. So we're working with our developers to build up these sort of trustworthy communities. We're working with a couple of foundations to pay patients to contribute their data to these communities and to put them, the payoff is sort of, there's, especially for women of color and sort of diverse patients with very poor outcomes, there is massive need for second opinions and matching the clinical trials. So that's our goal, to collect the data, organize the data, machine learn the data where we can actually put patients and physicians together in sort of a trusted space. So we're working on that now. And I think that's it. I don't have time to read these.
Thank you so much, Dr. Hadley. So I don't see question pop up. So, but as we are falling behind the schedule, so I do have some question for you, but maybe I can just, we talk from via email later on. So, so is that, so we are moving to our first presentation by Dr. Peter Robinson. Dr. Robinson, you can set up your slide. So Dr. Robinson's presentation title is GA4-GH Phenopacket Schema, a computable representation of clinical data for precision medicine. Dr. Peter Robinson is a professor and Donald Rooks Chair, Genomics Computational Biology at Jackson Laboratory for Genomic Medicine. Previously, he worked as a pediatrician professor for medical genomics at Charité University Hospital in Berlin, Germany, and was adjunct professor of bioinformatics at the Free University of Berlin. His group developed the human genotype ontology and software for translational genomics. Dr. Robinson, please take it away.
Good morning, and thank you to the organizers for the invitation. So I will share my screen, and so just set this to full screen mode. So before I start, I'd like to thank the many people with whom we did this work, especially Melissa Hendel, whose idea this originally was 7 years ago, and Jules Jacobson, with whom I had a very close collaboration in developing the protobuf code and a lot of Java code to work with phenopackets. So as mentioned, my group developed the human phenotype ontology, and we've had a lot of work here extending the ontology with the community. And as you can imagine, I've gotten a lot of messages from people like, here's some HPO terms for my patient. What do you think this might be? And people would send me a Word file, an Excel file, essentially lists of HPO terms without structure, which is very difficult to compute on. And in general, there hasn't really been a great way of recording the clinical manifestations of, especially of rare disease. What we set out to do in 2014 was to develop a standard for representing a case report, an individual patient with their phenotypic features, their measurements, and other things, which I'll show in the following slides. We developed the standard as a part of GA4-GH, and I'm not going to have a lot of time to talk about it today, but our standard interoperates with the VRS standard for representing variants. We're in the process of implementing interoperability with the GA4-GH pedigree standard, and we're working with the GA4-GH beacon group to enable basically an API, a REST API-based search using many parts of the Phenopacket standard. Yeah, I sort of mentioned this. Standards enable computation, and when I was preparing this talk, I just remember in 2010 we wound up publishing, I think, the fourth novel disease gene discovery by Exome, and so our group in Germany had one of the first Illumina genome analyzers. And we had to do all of this without any of the standards you see here. So there was no VCF, and the development of VCF enabled many other tools that we now take for granted. It took us about 3 months to find the disease gene in this case. And with the tools we have now, we could have solved this case in about an hour. And so our goal is really to set up not only the standard that I'm presenting today, but to start to inspire the creation of a library of tools to use phenopackets to enable novel kinds of computation for phenotype-driven research and genomics. So just to emphasize, there are a lot of standards out there, and we think that phenopackets will interoperate with some of these standards. I'll have a chance to mention FHIR towards the end, but really there hasn't been a standard whose primary use case has been to provide, a basis for bioinformatics, for computational decision support using open source ontologies. This is an overview of the schema, and so a phenopacket represents an individual. So I can't really point here, but if you can direct your attention to the black bar that says phenopacket, and look at the other elements that are related to that by arrows. Towards the left, you'll see individual who has a number of information such as IDs, sex, vital status. There's a disease element that represents an overall clinical diagnosis. I'm going to skip the interpretation element for a second. Go to the phenotypic feature, which essentially contextualizes ontology terms that represent phenotypes, including HPO, but you can use other ontologies if you like. There are clinical measurements that where one does not want to transform them into ontology terms. So for instance, if you have a potassium value of 3.0 milligrams per deciliter, that's hypokalemia, but perhaps you want to retain the numerical result. That would be a measurement element. There is a biosample that can be used to represent biopsies, tumor extirpations, anything related to a biosample. There is an element called medical action that allows you to represent medical treatments, procedures, including surgery, therapeutic regimens for cancer and other things. And finally, one of the use cases of a phenopacket is to relate this data to genomic data, including but not only VCF files of genome sequencing. And so we're have elements here that are closely related to the VRS standard that allow you to represent and interpret variants. This will give you, I have a few slides that will give you a feeling for how the schema works. So this, here we see two phenotypic feature elements. The first one represents a type of epileptic seizure, infantile spasms, but it's contextualized. So we're able to say that it's severe, that it occurs on a daily basis, this is quotidian, and that its onset was infantile, which is defined to be between months 2 and 12 of life. The resolution of this abnormality was at 4 years, 2 months. And another phenotypic feature, we see that it's excluded. So global developmental delay was excluded. And in fact, this two combination of observations is characteristic of some Mendelian diseases. Here is an example of a measurement where we're looking at the number of platelets per volume. And so this would be represented in the HPO as thrombocytopenia, so too few platelets per volume. But in this case, we're keeping the numerical measurement of 24,000, and the measurement message allows you to represent the normal range used at the laboratory where the measurement was made. Here, this gives you an idea of how we represent medical action, so therapy in general. And this is an example of a patient who died of COVID. And this patient had a previous medical history of a left ventricular assist device, which was planted in 2016. And here we're only showing the treatments. Other parts of the standard would show the rest of the COVID history, but here the patient was initially administered oxygen via nasal cannula at initially 2 liters a minute, which is not an enormous dosage, and a little bit later on increased to 50 liters a minute. The same patient also received essentially the Regeneron cocktail, which is shown on the right side of the slide. So we see a number of use cases for phenopackets. We have been developing a FHIR implementation guide that I'll show you in a bit. There are a number of applications that you can now use to generate phenopackets. We have an extraction from UMAP. We are talking with quite a few journals in the human genetics space about cajoling or even requiring authors to represent case reports using phenopackets. The use cases are many, including matchmaking, cohort identification, decision support, and some of the tools in the space for rare disease genome analysis already take phenopackets as input. We've developed a FHIR implementation guide and now have software that is, I'll say, finally working to essentially generate phenopackets, populate a server, retrieve it from the server, and translate it back to GA4GH format for use in downstream bioinformatics applications. We have a number of software tools that can be used to create phenopackets using, we have I think a very good text mining library in Java and several applications like the one you see here that allow you to enter HPO terms, data about genetic variants and other things to create phenopackets. So on this, the manuscript for phenopacket has just been accepted. We're distributing these slides to a number of internet addresses to where you can get the software. I'd like to thank NHGRI and NLM for the funding, and thank you for listening.
Thank you so much, Dr. Robinson. It's a wonderful talk. So there are 2 questions I see. So there's one question posted in the QA. Let me read it first. So will there be any tutorials or workshops on how to use phenopackets?
Yes, indeed. We are preparing a very detailed example as a tutorial of how to interpret and use all of the sort of features of a phenopacket that is invited by a journal called Advanced Genetics, and that will be available soon. We are going to be hopefully presenting this as I think it's been accepted a workshop at ASHG this fall that will hopefully take place despite COVID. Yeah, that's
sure. Yeah, great. Yeah. So another question is, so I mean, we have audience show interest in seeing how, so the power wash from Dr. Musin's presentation may like intersect with GA4 GH phenol package. So do you have a thought for that?
Yeah, I mean, I think that's a wonderful tool, and it definitely could be used as a pipeline to help create phenopackets. I think to make the best phenopacket possible, you would have some type of human intervention, but we're envisioning tools where you would use text mining to present a human curator with suggestions for vetting. In general, it would be wonderful to use tools like the one that Mark presented to kind of clean the data at source at Geo. So in part of my work, for instance, we've been trying to download 100 RNA-seq data sets with specific metadata. It's an enormous amount of work right now, and actually coding this data using various schemas, including phenopackets if appropriate, and with a core set of metadata would be so valuable for research. And I really wish that NIH would fund a team of curators to kind of clean up the various databases that we all use. It would save us all so much time.
Thank you so much. So yeah, and thank you again. So it's really a wonderful, great talk. So I mean, now we are moving to our last presentation. So Dr. Alison Marsden, please set up your slide and let me introduce you. So Dr. Marsden's presentation is automated data curation to ensure model credibility in the vascular model repository. Dr. Alison Marsden is a professor in the Department of Pediatrics and Bioengineer and Biocourtesy and mechanical engineer at Stanford University, where she is also a member of the Institute for Mathematical and Computational Engineer. And Dr. Marsden is the recipient for several awards, and also he has been elected as a member for several major societal scientific societies, including like AIMBE and SIAM. So she has published over 130 peer-reviewed journal papers and served as on the editorial board of several leading journals. Her research focuses on the development of numerical methods for cardiovascular blood flow simulation and application of engineering tools to impact patient care in cardiovascular surgery and research. So Dr. Marsden, please take it away. Thank you.
All right, thank you so much, Yan Li, for the nice introduction. So we're going to shift gears a little bit and talk about cardiovascular disease, and in particular, my lab and my particular area of focus has to do with bringing predictive simulations from more traditional areas of engineering. So we're used to the idea of using predictive modeling for things like aerospace and automotive engineering. And so we've been developing a set of tools to kind of bring a lot of that technology into the realm of surgical and treatment planning for cardiovascular disease. And we do that through a set of open source tools that I'm going to tell you about. And really the sort of backbone of this process is shown at the bottom of this slide. So we typically do image-based patient-specific modeling. So we start with either a typically MRI or CT data for a particular patient. And then we do image processing, centerline path extraction and image segmentation to generate a three-dimensional anatomic model for some portion of the patient's anatomy that's relevant to whatever disease application we're interested in. We generate typically an unstructured tetrahedral mesh, apply boundary conditions, and then we run finite element simulations of blood flow in that patient-specific anatomy. And we can do this for cohorts of patients. We can do test sort of different scenarios in a controlled manner. So what do we use these type of simulations for? I'm not going to go into the applications in this talk too much, but just to give you a flavor, for example, we can do controlled comparisons of different scenarios, such as different graft designs in coronary artery bypass graft surgery. On the bottom here, you can see pulmonary arteries of children born with congenital heart defects like Williams and Alagille syndrome. So these lead to severe stenoses in the pulmonary arteries, and we've used computational tools to compare different interventional, so stenting either the proximal and or distal pulmonary stenoses and looking at the effects. We can also do kind of controlled design optimization of graft designs, and we can also look at kind of the biomechanics or mechanobiology of disease progression, where sometimes the mechanical forces acting on, for example, the walls of a blood vessel are difficult to ascertain using traditional methods, and we're able to obtain them via simulation. So these are the types of things that we use these tools for. And my lab, together in particular with Sean Shadden's lab at UC Berkeley, has been developing and maintaining two key open science resources for the cardiovascular biomechanics community. The first is SimVascular. So this is a modeling and simulation platform that provides that entire pipeline from loading in your medical image data set to building an anatomic model and running a finite element simulation. And this is called SimVascular. The website is there. I encourage you to have a look at it. So this is a software that was originally developed at Stanford by Charlie Taylor. He used it to form the basis for HeartFlow, which is a new company that just got FDA approval a few years ago for assessment of coronary artery disease. And it provides an open source pipeline for this whole process, medical imaging to simulation. The open source package, which Sean and I kind of took over the maintenance of several years ago. We have about 6,500 users worldwide. We've been supported by the NSF through their SSI program since about 2014. We also have a gateway through Exceed for educational use, and we have students around the country who are using the software for things like class projects. Okay, and then the focus of today's talk is a partner project to SimVascular, which is called the Vascular Model Repository. And this was originally developed by Nathan Wilson under an NHLBI contract, and it's an open data resource that's compatible with SimVascular. It provides full data sets, full SimVascular projects that contain medical images, anatomic models, meshes, and finite element simulation results for a range of cardiovascular disease. And our goal is to support now, using this repository, new efforts in AI and machine learning, medical device design, reduced order modeling, and similar projects. We have a new website that's shown here. And I'll make the point here, so we have about 92 projects in the website currently. We're working on significantly expanding that, and I'm guessing over the next 6 to 12 months, this is going to be roughly tripled in size. These data are very sort of intensive to generate. They require a lot of user intervention in building these 3D models, and then significant computational resources to run simulations on each of these patient data sets. So running one of these simulations requires usually hours to a couple of days on a large computer cluster. So we are in the process of expanding this and opening to external submission. And then I'm going to talk a little bit today about our curation efforts in developing kind of software to ensure model credibility, and some of the examples of how we're using this data to drive various efforts. So what does this all look like? These are a sample of medical images that are included in the repository. And again, these are 3D volumes of CT or MRI image data stacks. And our vision is that users can kind of interact with this data at different points in the pipeline. So people may just be interested in the images. They can access those. They may be interested in the actual flow simulation results, which are all solutions of the Navier-Stokes equations. They can access those sort of at whatever point in the pipeline is relevant for their research. These are the just a sampling of the simulation results that are contained in the repository. And you can see here that we have spanned a wide variety of different regions of the cardiovascular anatomy and different disease settings. My lab has a particular focus in congenital heart disease. I think we have an unusual number of models and simulation results for some rare congenital heart defects, such as single ventricle disease, as well as more common things like coronary artery disease models and abdominal aortic aneurysms. But you can see kind of the wide variety of different regions of the anatomy. Some of that is summarized here. So these are the model types that are currently contained in the repository, spanning pulmonary, aortofemoral, aorta, congenital heart disease, coronary. We have a few animal data sets included, and then a number of different kind of disease diagnoses, some of which are rather rare, including Kawasaki disease, aortic coarctation, pulmonary hypertension. We have a number of models that are pre and post treatment, such as a Glenn to Fontan conversion for single ventricle patients. And our goal as we go through the rest of the expansion process is to also kind of round this out and fill in gaps where we may be sort of missing key areas in attempt to sort of span the space of cardiovascular diseases and anatomies as best we can. So for the rest of the talk, I'm gonna tell you about some of our efforts in using the VMR for curation, both in images, models, and simulations. So our data set is unique in the sense that we have these various different types of data that are linked and compiled together. So some of these efforts include using reduced order models for fast curation. So if we have a new data set coming in, we want to be able to very quickly run kind of reduced simulations on it and make some assessments. We've also used these reduced order models to actually accelerate our simulation pipeline. We've also used the VMR for training machine learning networks for faster image segmentation, for quantifying uncertainty in simulation predictions. And then for developing, I think it's going to be really critical to use this data, both the anatomic modeling data, which has this really valuable segmented ground truth data, as well as the simulation results that can inform physics-based machine learning models. So developing improved reduced order models. And then I'm not going to go into it today, but we've had significant interest from medical device developers. They're often looking for image data for a variety of different disease applications and looking at how their devices are going to fit into different scenarios. And so this is meant as a valuable resource for that community. So in the cardiovascular modeling world, if you will, we have in general 3 levels of different fidelities of models that we perform. So these span kind of less to more computational expense and less to more resolution. So on in the bottom corner, we have 0-dimensional lump parameter circuit analogy models. These run, very, very quickly, but give relatively little spatial information. In the middle, we have 1-dimensional models. These are PDE models that run relatively quickly and give 1 dimension of spatial information. And then the highest fidelity level, which is mostly what our focus is the full-blown 3-dimensional finite element models. So there you get very detailed resolution, things like wall shear stress maps on time resolved on the all of the walls of the blood vessels that you're interested in. However, these also span a limited piece of the anatomy because they're very computationally intensive. Okay, so one of the things that we've done is develop an automated pipeline to kind of, starting with one of these high fidelity models, automatically run lower fidelity models on the same patient anatomy. So you can see here an image of a patient-specific aorta femoral model with its various branches, and then an extraction of kind of just the branch pieces of that model for a reduced order simulation. So we have an automated way to split the 3D geometries into branches and junctions for this analysis. This was done by my postdoc, Martin Thaler. And then that allows us to carry out this, what I call 3D, 1D, 0D pipeline, which is fully automated and available in SymVascular, so that you can take the data in the repository and use our open source software, SymVascular, to kind of run these reduced order simulations. So what we do is we start with a 3D model in the repository or that somebody has generated. We do centerline extraction with VMTK libraries. We can extract then the vessel segments as well as the junctions. And then we can use SymVascular's 0D solver and 1D solver to run fast reduced order simulations. This is an example of 2 different cases where in the top case, in the healthy aorta, the reduced order models both perform very, very well. And later in the talk, I'm going to talk a little bit about improvements that we can make using the VMR data to actually improve the reduced order models themselves. But you can see here in the healthy aorta that both the 1D and 0D models actually perform very well, and this is somewhat expected. But in the bottom case here, we have a coarctation of the aorta with a severe stenosis, and here is a case where our 0D and 1D models actually break down and don't adequately capture the pressure drop across the stenosis, and that's of course due to the nonlinearities of the blood flow. So we've actually run this automated pipeline on the entire vascular model repository, about 100 models, and this allows us to kind of classify, well, where are we doing well and not well. And then we've also been able to run convergence checks using the reduced order models, and this is useful both for assessing kind of convergence of new data that might be coming into the repository, as well as for accelerating our own simulations. And this is just using the kind of properties of the typically the RCR circuit boundary conditions that are used at the outlets of our models. And again, we have an automated pipeline that's allowed us to analyze this convergence on all of the data in the repository, as well as new data that's coming in. And using this pipeline has allowed us to save quite a bit of computational cost in using reduced order models to actually initialize 3D simulations, saving in some instances hours of compute time through this automated process. We've also used the repository for deep learning to automate our image segmentation process. So extracting from about those 100 models that I showed you, we've extracted about 16,000 two-dimensional slices, which we use to train a deep learning network using vessel regression. And what you're seeing on the right here is DICE scores for large vessels, where we're doing particularly well in predicting the resulting segmentation compared to the ground truth. And then this breaks down a little bit for smaller vessels, less than about 0.4 centimeters. But we're working on ways to further improve that now. But this saves an enormous amount of time for users, and this has also been released as part of Synvascular. As part of that, we wanted to also assess uncertainty. So these simulations have enormous uncertainty that goes into them from, mostly from sources of clinical data and medical images. So you can imagine the process that I just talked about with image segmentation. There's noise in the medical images that needs to be accounted for. And so we've developed pipelines for doing both parameter estimation under uncertainty and also propagation of uncertainty to simulation predictions. So in the setting of image segmentation using the repository, we've used Bayesian dropout networks to produce actually families of segmentations that we can sample from, and we can propagate those, the uncertainty from that image segmentation process that comes straight out of the machine learning network. We can propagate that all the way through to the finite element simulation results and actually put confidence intervals or uncertainty bounds on the simulation predictions. You're seeing that here. This is a plus or minus 2 standard deviations on pressure, shear stress, and flow rate predictions in an abdominal aortic aneurysm model from the repository. And again, this is only the uncertainty coming from the actual medical image segmentation process. There are a number of other sources of uncertainty that we've accounted for in other ways, from the boundary conditions, material properties, et cetera. And then just to kind of briefly wrap up, some new areas that we're interested in have to do with using graph neural networks to develop new reduced order models to try to improve on some of that discrepancy that you saw earlier in the 0D and 1D setting. And so here we're actually training from simulation data from the VMR and then using the graph neural network approach as a solver. And doing reasonably well, still working on improving some of the generalization error in this setting, but have about a 10-second rollout at this point, which is quite fast. And then we've also used the VMR data, so extracting about 150,000 junctions automatically. So every time we have a vessel bifurcation, and then training a neural network to predict pressure loss across all of the junctions in are using the data from the model to predict pressure loss in those junctions, and then have achieved improvement over previous empirical models that are standard in the field. Okay, so I'll wrap up there. I particularly want to acknowledge Daniele Schivazzi, Sean Shadden, and Eric Darve, who have been instrumental in collaborating on UQ, machine learning, and Synvascular, as well as clinical collaborators who've generously helped us obtain clinical data, and in particular, our software engineering team, Dave Parker and Nathan Wilson. Thank you so much, and I'm happy to take questions.
Thank you so much, Dr. Marsden. So I don't see question posted in the QA, but I see someone is sharing information in the chat, so we can all take a look at that. So I have a very general question. So what are the types of users for this repository? So clinicians, researchers, or
yes, all of the above. So I would say cardiovascular researchers, clinicians, so clinicians who are interested in treatment planning, both surgical planning and other kinds of interventional treatments. medical device developers. The FDA is increasingly interested in using simulation data in the medical device approval process. And oftentimes it's difficult, particularly for medical device startup companies who have more limited resources. We've been contacted by several startups in the med device space who are interested in different types of data. We're developing a device for pulmonary hypertension, and we want to know if our device is going to fit in different types of patients. And so being able to provide that type of data is one of our goals. Yeah.
That's really wonderful. So there are other questions, but we are, running into the second session. So thank you again for our wonderful speakers, and thank you for all of the audience to stay with us. So the next session will start at 11:55. So I apologize. So we run over the schedule a bit. I wish a success for the next session. Thank you all.